Very Swiss here. I was just given a sign by the audience that we should start. Dear guests, dear colleagues, a warm welcome to tonight's Novartis campus lecture. What an honor that Dr. Katalin Karikó has accepted our invitation to come here to Basel and be with us, spend the evening with us. And what an honor also that I'm looking at a near full audience on a Friday evening, 6 o'clock. Very impressive. As per tradition for those lectures, those of you who are regulars will know, we have a Novartis representative to give a short welcome address before we start the lecture. Again, we're very honored that Mr. Reinhardt, Dr. Joerg Reinhardt, has accepted this role. He's here with us this evening. Now I have two more invitations, and this time I'm directing them directly at you, dear audience. One is, there will be a Q&A, we call it the conversation. So at the end, I'm inviting you all to start thinking about questions. This is your one chance to ask Katalin Karikó a question on stage. The second invitation is very important. The philosophy of those events are always to merge encounters with learnings. So you're all warmly welcome to join us for an apéro riche right after this lecture. Very important. Now, that was it. Without further ado, Dr. Reinhardt, the stage is yours.
Good. So, ladies and gentlemen, and dear colleagues, also for me, it's a big pleasure to welcome you to the Novartis lecture, which gives me the opportunity to introduce you to Nobel Prize winner Dr. Katalin Karikó, whose contributions to science and medicine cannot be overstated. Without her decades-long work on RNA technology, we might still be frozen in a lockdown and struggling to organize our life and work from home, always in fear of contracting the coronavirus. Rather than coming here together at the Gehry building, we would still be holding Zoom meetings to get a sense of togetherness. I remember that shortly after the outbreak of the pandemic in early 2020, there was little hope that the world would find a quick fix for the outstanding healthcare challenge. Although many companies, including ours, tried to repurpose existing drugs, it was almost to no avail. Most therapies could only soften some of the symptoms, but were unable to tackle the virus itself. At the same time, the prospects looked grim, given that development times for new drugs and vaccines are counted not in years, but very often in decades. While it takes 10 years on average to develop a new drug, average development times for vaccines can be even much longer. It took almost 40 years to develop a vaccine against chickenpox and over 40 years against Ebola. There is still no vaccine for AIDS, MERS, or Zika, even though researchers have been working on them for years and years. As the world was frantically looking for a solution, Dr. Karikó, unknown to the general public, finally succeeded in speeding up her work on an RNA therapy at BioNTech, which she had pursued throughout her academic and professional career. We all know the story and remember the great relief when we heard of the first successful vaccine tests, which came in record time and presented the world with a novel technology to save us from the grip of the pandemic. However, in the first rush of celebration, the work of Dr. Karikó was eclipsed again as the public was debating the pros and cons of RNA technology, which most people were unfamiliar with. Skepticism and conspiracy theories mushroomed all over the place while the pundits discussed the challenges of production and distribution. It was not until the New York Times in 2021 featured Dr. Karikó's work, shedding light on her role and that of her colleague, Dr. Drew Weissman, in the rapid development of the vaccine. It was the first time that a major news outlet shone the spotlight on her work and retold her meandering and at times frustrating scientific and personal journey. It was a narrative of personal hardship and sacrifice, yet always held together by Dr. Karikó's love for science and her deep belief in the therapeutic potential of RNA technology, which for years had been viewed skeptically by academia and industry, including Novartis. She knows what I'm talking about. In many respects, her life and career best exemplify what it means to be a researcher striving for true novelty. While most scientists aspire to find something new and exciting and secretly yearn to be recognized with a Nobel Prize, what connects the best scientists is courage, perseverance, and an unyielding belief in their own hypothesis. From this vantage point, Dr. Karikó can teach all of us what it means to work diligently on a project, not for years, but for decades, finding the right collaborators and mentors and never giving up, even if the situation gets darker by the day and criticism piles up like a mountain. As such, she is a role model for all of us, as we need the same perseverance and focus on excellence when it comes to our own projects that we believe in. With this, I would like to close and hand over to Dr. Karikó. Thank you.
Thank you. Thank you very much. Can you hear me from this one? Yes. Do I need this? No? No? Okay, thank you. Dear guest, it's unbelievable that I am standing here in front of you. And last time it was December, 650 people watched me to give the Nobel Prize lecture, and I was so nervous seeing there. And as we talk, I remember it's not the first time I am at Novartis campus. I was not here at that campus, but in Boston. It was 12 years ago. That was, for me, it was unbelievable experience because nobody knew who I am, and I was expecting maybe I get a job offer. Not happened, but anyway, I went there because Novartis was the first company who started doing messenger RNA. Yep. And then I expected that, I can work there and doing other things. But anyway, I revisited 2014 with Uğur Şahin, the chairman and the chairman of CEO of BioNTech, because I told him that Novartis is a great company and they are working on RNA. So that's what comes to mind about Novartis, and this is wonderful to be here and beautiful campus, and thank you very much for inviting me. So I learned that the audience will be, some scientists, some children, and some forever young scientists, and so I have to prepare accordingly. And so the people, when think about what scientists are doing, maybe that magic which is here, thinking that hocus pocus is happening, and I want to tell you about a little bit my life and tell the young one that how they can be maybe scientists. I cannot guarantee that what would be the outcome, but that's for sure that all children are curious, and that was I was. I grew up in Hungary, and a very simple household. My parents had just elementary school education. We had garden, and I remember, the chicken house coming out from the eggs, and the neighbor had a cow. We had to go there to get the milk. But one day I have seen how the cow delivered, a little cow, and how they pull out, and this little animal was there. So many interesting things happened there. We had gardens, and we, with my sister, we put the seed. Our parents, give us that part, which usually not very useful land, but anyway, some plant came out, and we learned. And every child is curious and want to know things, and I want somehow to make sure that they stay curious and they want to learn, and so I learned a lot from my parents, as my father was butcher, and I was told that I was watching how he opened the animal, and then I was watching that what is inside, that animal was running and now it's not running, what it made running. And I don't remember these things, but I learned from my parents that hard work is part of life, and then how to make sausage, which, sometimes, you need in life, some job to maintain. And so in school, my sister was 3 years older, so, if you have a sibling and she's good in school, that teacher's expecting you have to be good. So that's why I studied very hard. And I started to compete with different kind of competition, geography, chemistry, but biology I was the best, all the way that in elementary school I was third best in Hungary. And so I was just in this small town coming and reaching that kind of level. Interesting to mention that this was, we were behind the Iron Curtain, and there, for this elementary school children who local competition, biology competition, they succeeded, they organized them summer camps. And here there is the tallest girl standing there. But here, these other girls later I met in life became physicians, chemists, biologists. So it is important that even, you are from high school, but elementary school children should be here, because somehow even elementary school children already knew that they want something. And so it is important, to maintain their interest and work with them. I was lucky that in this small town had a beautiful high school, and I had great teachers, and they maintained all of the interest and showed all of the experiment, chemistry, biology, and so my teacher, Albert Todt, said that I could be a scientist. Of course, there is just 10,000 people lived in this little town, but I believed him. So everybody needs a cheerleader, and he believed that I could be a scientist. And I didn't really know what scientists. I was reading about them. I learned that they discover vitamin. That would be, a big thing at that time. And so as my parents were not highly educated, there was in this system, in the communist system, that I could spend the summer, like 2 weeks, 1 month, in a camp, which was in middle of the July, and then from early morning, late evening, I could go to the university with other children who were, underprivileged, and study, so catching up in biology, physics, chemistry, mathematics, a lot of mathematics we studied. So it was organized by the university. If there are students from the university, I am giving them idea how they can help the system. So on the very right, that's me. That was cut out from a newspaper which reported that. So why I went to this University of Szeged, which organized this, because my dream was that one day I will come to this University of Szeged, because in the same year the Biological Research Center was opened in the same city. And that was my dream, to work in this Biological Research Center and discovering things. I don't know what I had in mind at that point, but as a student I immediately started to visit and try to go get a position there, like a student assisting something, and chemistry and genetics was the most interesting, but for me, the only place was their, lipid team. Lipid is grease, not really very exciting, but Tibor Farkas, who was heading this institute in this section, telling me about that the membrane, actually how I learned in high school, the layers of protein and lipid, is not true. It is different, and it was immediately started to be excited. So I told him that I would like to work there during the summer. I was 20 years old, and he said that, rest of your life you can spend on the bench. At that time, I didn't know that at the age of 57, I'm still doing my own experiment. So what he was right. So he said, just go to the Fishery Institute and then collect fish fat from this farmed fish, and we are analyzing the composition because these fish were fed with different kind of chow material, and we have to see that whether if you give, let's say, corn to the fish, whether it will be the fat like the pig fat. And then he said also that collect some water fleas. So that you go to the university, dream about big genetics, chemistry, and finally, you are picking up water fleas. And that's what. But what is important is that you have to always accept it, go with the flow, and learn as much as possible. Let's say the water fleas, or those lipids you have to measure and decide that whether in the future you will consume any farmed fish material. But anyway, so you learn a lot, and that's what it is important. So I hardly could wait. Summer is over. I go in a real institute, which a real science at the bench. And so when I went back in the fall, that time, two scientists, Ernő Duda and Eva Kondorosi, approached our team that they need some phospholipids because they would like to put some nucleic acid into it and deliver to the cell and make liposomes. And I just thought that this is a great idea. Everything was new for me, of course. But we were behind the Iron Curtain, so we couldn't purchase that phosphatidylserine, phosphatidyl ethanol amine mixture. But Ernő found a paper from 1942 that you can isolate from cow brain. No? Came handy that I am the daughter of a butcher, so I was not gooish about working with a big bucket of brain, and we spent the whole week, and really we isolated these lipids, and we could do this experiment. And one day, so I was still a student, and one day Jenő Thomas walked in, and he said that he has opening in his RNA lab. So don't think that I have any special visionary thing that know that there is a formulation, there are lipids, there are RNA, and one day all of these belong to. Yeah, I just went with the flow because I want to have a PhD. And here, so again, I dig into it, this process. And that was 1978, and 77 discovered this small 2', 5' oligoadenylate, a short RNA molecule, which had antiviral effect. And Jenő was just coming from the industry, and he said, we need an antiviral compound. It was 1978, even today we need, isn't that? We need an antiviral compound. So my job was to synthesize chemically, enzymatically this small molecule, this small RNA molecule, set up a viral screening lab. All of my colleagues were organic chemists, and so it was a big job for me. And I learned how to work with this molecule. And of course, this molecule is actually naturally made in your body. There is this enzyme, oligoadenylate synthase, written there, OAS, and it activates an enzyme, which is RNase L, and that was the idea that cuts up the viral RNA. I have to say, those who are at the beginning of their career, you just keep following what happened on your field, because I have to say that science identified that these are the two enzymes which is very low level present on those children who ended up after coronavirus infection in the hospital, intensive care. These two enzymes, and because the polymorphism-related changes resulted it, and nobody knew about that their level is low, but when they measured all over the world what was the problem with those children, these two enzymes was responsible for it. So anyway, we work on this project for one pharmaceutical company paid for the project, but they could see that it is a difficulties, to getting this small molecule in. It was antiviral, but getting to the right place. So I was 30 years old, lost my position, and ended up in the United States. I would tell during my talk that I was terminated in my position 3 times. I was demoted from my faculty position, and I wouldn't be here. All would not happen to me. So don't be disappointed. It's not guaranteed that things going well later on. But so I ended up at Tampa University, where also with Professor Suhadolnik, we worked on this small molecule, this 2', 5' link molecule. And there was a big problem. We are in 1986. 85, because there was HIV. And we needed antiviral compound. We couldn't make it formulate in a way that it could be useful. So what we did, we did double-stranded RNA. And we try to induce the natural interferon system in 20 patients. We treated HIV patients with double-stranded RNA. And if you paid attention, I mentioned single-stranded RNA, RNA that, cutting up RNA, labeling RNA. Now it is double-stranded RNA. So I am here, at that time, 34 years old. Everything was in my life about RNA, labeling it, cutting it, changing it somehow. And in the field, I was not working with mRNA yet. It has to come. And I have to say that there are the major milestone of mRNA, messenger RNA development. mRNA, we just say, in science, in short, it is messenger RNA. A messenger RNA is not something we invented. It is present in our cells, and it carries information. It was discovered in 1961, and scientists discovered many characteristics about it. This is how usually science happening, and then 1984, first time they could make RNA in a test tube, and then the scientists thinking that what this is useful, and started to do experiment animal and try to find some usefulness for this. And from 2000, when companies started to form, and they try to do it use for human trials. So that's what I am talking about somewhat. So the information flow was established with this one, that there is a DNA which was, at that time, 1952, was well established. It is in the chromosome, it is in our nucleus, it is very stable. And then all of the protein, which we are made up, is happening in the cytoplasm. But for quite a long time, they could not find that how the information gets to the protein synthesis factory. And that was because this messenger RNA was so labile. And At the beginning, a scientist couldn't synthesize it, so they just could isolate. And I mentioned just one isolation because in the red blood cell precursor is full of globin protein. And some scientists could purify it. It's the precursor of these red blood cells, the reticulocyte. And then they put it in liposome, delivered to a cell, which were isolated from mice. And then this cell could make that protein. And this was 1978. So 1978, already scientists could make, isolate an RNA, would make a protein. So the things, the RNA development, messenger RNA development did not happen overnight. A lot of overnight was there, and a lot of discoveries needed, and a lot of technological. I like this thing, what Sidney Brenner said, that progress in science really starts with new techniques. Because, of course, I did a lot of technical things at the laboratory. And then these techniques opens up new possibilities, and then you do new discoveries. And this is the way. So scientists at Harvard University discovered that quickly you can make RNA in a tube, in a test tube, by using phage polymerase. The phage is a virus of the bacteria. So the bacteria also has to worry about viruses. And so then you can make an RNA. They made actually interferon coding RNA. They synthesized using this RNA polymerase. And then when they inject it to a frog oocyte, if you are old enough like me and you are scientists, you know that in laboratories usually there was a terrarium and there were some frogs there because they needed eggs, because those are large cells. So at the beginning there were no culturing cells. And then they injected to these frog oocytes and cultured, and the medium contained the human interferon. So that was in 1984. When first time synthetic RNA was injected in living cells and produced the protein which was encoded by this RNA. So you can imagine how scientists get excited at that time. And they injected RNA everywhere in mice, and then they could find that it was translated, and RNA was already used in the early 90s as a vaccine for infectious disease, and also as a cancer vaccine. And I was also started in 1989 using messenger RNA. And when you would look for these scientists and these papers, I was like following Yerikovsky's paper because I was interested to use the messenger RNA for therapy. So you look at there, and they never ever published ever again anything about mRNA, and many of these scientists. So now that recently I get more known, so I asked them why they didn't do it, and they said that they could not get funding, and that's why they left the field. And of course, many problem was with RNA. Mostly RNA had just problem. One is that it was so unstable. It was very small amount of protein, was made very short period of time. So it was considered only just a laboratory technical material and not for any other therapeutics. Anyway, so I ended up in 1989 in cardiology at Temple University. I was terminated. I missed that part. And then I ended up here at cardiology department, and I started to work with Elliot Barnett, and he was interested in blood vessel. He was working with catheterization and bypass surgeries. So I tried to see how RNA could fit to this one. And so I worked with different blood vessels and realized that if I drip through RNA in a blood vessel, in 15 minutes later, the protein is made in that blood vessel. So I had to figure out what kind of protein this RNA should code that would be beneficial to survival of the bypass surgery. So we were working, and one sample I'm showing here, this urokinase receptor, which he was interested. And what happened is this protein is so complicated, so decorated with sugars and carboxy terminal, GPI link, and so on. And we put the RNA to the cell, and the cell knew what to do. Put there all of this decoration and made a functional receptor, which bound to the urokinase. And at that point, we knew that the RNA will be good for something. Maybe for bone transplantation when cell is in our cells and we can manipulate. But yeah, that's happened that Elliot left. I was demoted, and then came David Langer for my rescue. David Langer was a medical student in Elliot's lab. So I'm telling you, young one, that be kind to all of the fellow students, because one day, you know, they can help you to get a job. So that's what happened. And this laboratory I worked for 17 years, and David made sure that I will be not kicked out from there. Eventually I did get, but it was late. Anyway, my budget was low. You can see no lab coat, no safety glasses or gloves, but I worked with RNA. And anyway, with David, we figured out that now that we will come up with RNA, and we will treat a stroke patient, and we figured out how we can do it. So invention can come that you respectfully talk to a colleague who is in different field, understand the problem, you know what you can do and what he can do, and together can do something that nobody else. So sometimes invention comes that a lot of people working together on different angle look at things, or sometimes it is enough just two person. But yes, but David got a good job, and he left, and I met Drew Weissman at the Xerox machine, really. A lot of people don't know what Xerox machine is, or maybe here. Anyway, so what happened is he was working in the Department of Medicine. I was already a neurosurgery, but neurosurgery did not have the Xerox machine, so I still knew the code for Department of Medicine. Anyway, so he was the new guy there, and he told me that he wanted to develop vaccine, a prophylactic or therapeutic vaccine for HIV. And he came from the Fauci's lab, which at that time was not ringing any bell for me. And so I immediately offered that I can make RNA, and we agreed right there that we will work together. And so we did the first project, and as a result of it, we could measure that a lot of inflammatory molecule was made when we put the mRNA, the messenger RNA, which I thought that exactly the same good RNA which is inside our cell, we put immune cells, human immune cells, and they made this inflammatory molecule and did other things. Drew was happy because he wanted to do vaccine, and he thought that this will be a good one. But I want to develop a therapy for stroke patient, and inflammation is the last thing you want to give them. So we try to understand why. All in our cells we have mRNA, and it is not inflammatory. Could it be that it is coming from outside, and that was the danger? And so we were just curious to know that all of the RNA, if it is coming out from the cell, can activate and cause inflammation. And so that we decided that I will isolate a lot of RNA, and we will test out. And that's what we did. And what we found is that when we this purple shows the RNA, the beautiful RNA I made, and all of the other, the blue one which I isolated, you can see that those are not as inflammatory. And one of them, the tRNA, which was known have a lot of nucleoside modification, was not inflammatory at all. So that gave the idea that could it be that modification makes the RNA non-inflammatory? And to decide that, we just have to make mRNA with all of this modification. I just show you this, just those who are not in chemistry just scare a little bit that there are more than 100 different modifications are there, and which one we should do, and how our nature in our body is putting those modifications there. It was not known. Only 2 modifications was known, and that was my fellow scientist from University of Szeged. We lived in the same dormitory. Again, be kind to those together. Because when I called him up, asked for help, that he responded to the telephone. And so he discovered how 2' methylation and how pseudouridylation happening in the ribosomal RNA. But at that time, it couldn't be reproduced in a test tube, so I have to call up János Ludwig, who I was a graduate student, together, again, be nice, and he told me that I could purchase modified nucleoside triphosphate and try to incorporate into the RNA. And that's what I did. I purchased what was commercially available, and I could make mRNA, which contains these 5 different modifications. And when we tested out on these modified RNA on cells, we found that now TNF is an inflammatory molecule. We found that not all of them induced inflammation. And when we look at closer, we found as long as the uridine was changed, then it was not inflammatory. Who knew that uridine was causing inflammation? Nobody. Of course, we want to make an mRNA which will be used as a vaccine or as a therapeutic, and for that, it was important that it will code for a protein. So when we tested out, we were surprised that one of them, called pseudouridine, not only was as good as the uridine, but it was produced 10 times more protein. I don't talk about those 2 years I spent to try to figure out why, but I show you another nice picture, for example, that when we put the mRNA coding for a protein which generates this green color, we could put in any cells, neurons or any kind of cells, and we could see the protein was translated. And you know that in the United States in that time, people are writing patents and establishing company. That's what I did. That was my company, RNA-RX. Never heard of. Yeah. Although we get $1 million from the US government because we proposed that we will make an mRNA which codes for erythropoietin, which will that who is following the bike race knows that increase the red blood cells, endurance, so on, so that we promise that we will demonstrate that messenger RNA can be useful, of course, for anemic patients. And so that's what we did. We injected mice with erythropoietin coding mRNA, which was nucleoside modified. Those are the red line. And we could detect for 5 days the protein circulating in the animal, whereas if we injected just the protein 2 hours later, it degraded. So, and it was functional. It increased the hematocrit value, shows the middle panel there, and it was not inflammatory. So everything looked good, but I was terminated in my position, and then ended up at BioNTech. So again, not when you are terminated and you kicked out from your position, it is always other people's decision. And what you have to focus on, what next? Don't waste time to compare that you were better than the other who is still staying, or not worse. You have to focus on what you can do, what next. So next I ended up in BioNTech. I have to say I was 57 years old, something like that. And the biggest achievement I felt at that point, that this old lady get 2 job offers, one from Moderna, one from BioNTech. And So I took the job at BioNTech, had no website, and I don't speak German, and it is far away from my home. But anyway, with Ugur and Özlem, we could work well together, and we further improved the RNA, made it, modification, purification, and so on. And end of it, we could get more protein, a longer period of time, so the messenger RNA was ready for prime time. So why I'm saying this mRNA is so important? Why? The mRNA is important because more than 100 protein product is FDA approved. And many of them is not, you cannot afford them. They are so expensive. And if you would replace the mRNA with mRNA coding for that protein, your body can be the factory to make that protein. So that's why it is very important. And of course, even if the final product is a protein, screening is always much faster to do with mRNA. And so many advantages, and it is very, very equalizer because it is affordable. That was very important for us. And when with Drew Weissman, we did not actually want it to license because we were naive and we thought that everybody can use it. But we learned that nobody will invest, nobody will spend any money if we don't have a license, a patent, which would protect their investment. And so that's why it was patented eventually. But so I am now is BioNTech and geared up for a new project. One of them I am mentioning here is we generated messenger RNA coding for bispecific antibodies. These are in clinical trial doing very well. I have this reference there. And here we just tested out on animals. This is bispecific antibodies. One part sees the immune cells, the other is the tumor cells, and brings them close proximity and helps the process. And so we did these experiments and many other experiments. I was also charged in this mRNA-based protein replacement program, and we did other things. We also collaborated with colleagues with Drew Weissman, invited them, and Norbert Pardy, my colleague, who already started to do this kind of lipid nanoparticle formulated modified mRNA project, and demonstrated that he could vaccinate very well monkeys. What was important is that the amount of RNA which was required for the mice was the same for the monkeys. A monkey is 4 or 5 kilo, you might not know that. But so anyway, in previously when tried to use DNA as a vaccine, they unproportionately had to increase the dose. So it was finally, the DNA, when in human trial, it was milligrams DNA were injected. But here, it seems that the same. We learned, of course, even for human is sufficient 30 microgram. So this was a Zika, because in 2017, the Zika virus was a major problem as some infection and spread there in Brazil. So that already this result was very promising. The formulation was lipid nanoparticle, and it was tested out later in human as well. So most of you familiar, you were sitting in the front of the TV watching this, what's going on during the pandemic, and in January we learned the sequence information about the coronavirus, and every technology was required for that, I have to say, because Yang Yongzheng, who sequenced a scientist in Shanghai, he sequenced and he put into the gene bank, but nobody looked at there. So he asked a colleague to put in the Twitter that the information is there. That was 2 days delay. But so even Twitter needed at that time, so the information will spread. And in January we learned about the sequence information, and in March, Moderna already started vaccination. for volunteers. And the people thought that, oh, it was just discovered and just all of these things happened so quickly, impossible, because those people were not attending to our yearly meeting, mRNA therapy meeting. They would know already that other mRNA vaccines for infectious disease were already in human trial. Moderna, CureVac already run trials. And those were, as you can see, 200 people, not 200 million, so that's why they didn't know about it. But there were already experiments and results. And where we are stand today, right now, more than 250 mRNA human clinical trial is ongoing. Many of them for vaccines for against viruses, flu, RSV. We already just heard Moderna get already the second RSV was FDA approved. Zika, herpes simplex. And there are also bacteria. So tuberculosis or Borrelia, which caused the Lyme disease, already in human trial. And parasite like malaria that the Gates Foundation is sponsoring this human trial. In addition, you can also hear about cancer vaccines. Both Moderna and BioNTech did a very successful trial running, and acute diseases, rare diseases, and more to come. And what I would like to say, because I can see that I am expired here, that to tell you that scientific discoveries is always coming, academia. We are thinking about discovering something, whether it is, how Cas9 was discovered, something in the bacteria, and how it will end up eventually in a human trial. So biotech companies formed, they testing out, they don't have enough money, so we need a company like Novartis or other company, large company who have run clinical trial. And eventually, the good news is if we get an FDA approval, which we thought that's the end of it. But during the pandemic, we learned that we need society to accept what is approved. So that's why I am here, and the colleagues here, the reporters here, that we have to educate the public. It is very important because, many of the people who were educated in immunology, in Facebook, and people who knew less, usually more confident about what they know. And so, therefore, we have to help. We have to educate the public, and they have to understand the human nature was always like that. They don't understand something, and they question, and something is new, and they blame. I took out that as a last picture here. It was more than 100 years ago when a publication came out, and of course that was the diphtheria vaccination, and they concluded that it is a human nature that we are not grateful for that we were protected from the disease. Rather, we will blame everything after happened the vaccination. And so that's even 100 years ago. In these days is also, you heard about people get syphilis, some get pregnant from vaccination, because prior to vaccination, they were not. And they did not know that something else has happened. So I can understand that there are real side effects because the vaccine is not inert, and all of the medical product has other effects, and the people had different kind of ongoing infection and inflammation and other things ongoing in their system, and of course it can be beneficial or maybe getting worse when they get the vaccine. But we have to educate the public about that. And finally, what I am doing now, what other people are doing actually mostly, because these are books for children about me. And is already published in different languages, and the right one is book I wrote about my life, and this is already was out in French in a week ago. In German will be August 21, and Chinese, Japanese, if you speak that, you can read already, and there is also Hungarian, and there are certain different languages, which I try to tell my story and something about, you know, the new generation. They are sitting here. That, you know, that how they can focus, how they can enjoy life and work, and have healthy the body, as well as learn how to handle the stress, because that could ruin them. But I give a lot of advice for that, and so hopefully they could learn, and then we will be in good hands in the new generation of scientists here and there. And so with that, I absolutely expired now, my time, and thank you very much.
Have a seat here.
Okay.
What a fabulous, inspiring lecture. I think I forgot everything I wanted to say because I was so in awe. I was literally in the story. Now, while you think of the questions, there will be 4 people with microphones in the room. I have one, obviously. I steal this moment to ask my question. No, but seriously, I mean, we learned science is not always glamorous, for sure. You showed that also with the pictures. We also learned that science can be highly entertaining. We learned that we should all be nice, kind to people, everyone. We should be curious, and we should be persistent. On the persistent part, I mean, it strikes me that your independence of recognition from the outside world and your rejection handling, that's almost like a superpower, it strikes me. And in another conversation, you shared your love for the Stoic philosophy. How did you come across it, and what do the Stoics give you in everyday life?
So when I was in high school, that teacher, Albert Todt, gave me this book written by János Seje, who was Hungarian. We couldn't speak, we learned Russian and Latin in high school, so we couldn't speak English, so we sent them Hungarian letter, and they responded. So that's why I feel constant pressure when somebody is sending me, now that you are listening, so many people, that I feel obligation to respond, because it was very important for us that they responded. And then we were so excited getting a letter from Canada, from Hans Scheyer, and then we read the book, and then we discussed how handle the stress. That in the book I described that, you know, he said that when you are very angry, upset, the quickest way you can cool down, you know, revenge. But he said, don't do that. It hits you back and escalates. He said, think about that, how you can feel gratitude for the same person you were just about to kick their butt. And then you have to focus, because then you can calm down. In my talk, I forget to mention all of these things. You know, I mentioned that without my teachers, without my parents, and without my wonderful husband, who is here with me, who wouldn't happen, because he was... Yes, because he was the one, you know, I went to Germany and commuted, and the first time I went there, and almost every night I cried. Am I stupid? I am 58 years old, I have a home, everything, and I am here in Germany in an attic, because that was the only place I could rent. And I am here. Every day I dress from a luggage. Now these days I used to do that again. But anyway, so, and what am I doing? Just the airport is there and I can go home. And when Christmas I went home and I told my husband that, oh, this is difficult. He said, I didn't try enough. So he sent me back. And of course, also, when it was the pandemic and stayed home a little bit longer, almost a year, one day he asked, don't you have to go back? Don't they miss you? And I said, aren't you happy that I am here? And he said, yes, I was always happy you came home and when you left. And I didn't mention about my daughter, you know, our daughter, who is a two-time Olympic champion, and who gets several awards here in, when she was competing in rowing here, also in Switzerland. And yeah, so we are a family.
Maybe there's also something in the genetics.
Yeah.
Just guessing.
Yeah.
Wild guess.
Yeah, actually, once we sent in 23 and me, because that's what my daughter gave us for Christmas. And it came out that my husband could be an Olympic champion.
Oh, ha.
Yeah. Not my daughter, but yes. Something genetic.
I only learned that I'm 10% Greek. I was very happy with this result. Now we're opening up for questions from the audience. Who dares to go first? Yes.
Good afternoon. I work at Novartis. I'm a Novartis person. I work in P&O, in HR. Never felt such a passion for a non-HR person, including scientists. And what I would like to highlight is that I think your story is a story of success, but also of rejection. So I think if I counted it well, you mentioned at least 4 times, I was demoted, I was rejected. And you made it fun, but I think in the moment was pretty painful. And this is probably something that several of us went through, are going through, or will go through. So do you have a secret recipe to manage that in the moment? Secret recipe that maybe is not a vaccine or a medicine. Thank you very much.
Yes, I agree with you. You know, I was not jump joy when my stuff was in the hallway 10 years ago and went to, you know, from coming Japan, I get a little red carpet event and I found my stuff on the hallway and throwing away everything which was there, a technician, and I, you know, she said, go to the chairman, decided that we vacate your place. So I was, you know, very unhappy, but and then really working 24 years, I walked towards my car and nobody said goodbye, hello, or goodbye party, just 24 years over. And so if you look back, then you would be cry, you know, that how I was treated. And people, I heard, oh, the university is not appreciated. University is actually, I don't know, there are some from, so these are just buildings, you know, and administrator, so that somehow you have to yourself value yourself, and you have to have the confidence and that feeling that what I am doing is good, and find a way how you can follow. Because, you know, if I don't have a grant, and I never had, at least I could convince another person that what I am thinking is good, and gave me salary. You know, my salary, I started at $40,000, $40,000 a year, and then 2010 I was up to 60,000. And the health insurance, the parking, what I had to pay is exponentially grow. So I get less and less salary, and it was not important for me, you know, because anyway I had no time to get hobbies, spending money. So So that somehow, I mentioned in my book also when my Russian language teacher, after I get, valedictorian, in high school, and just ready to leave, and he pulled me over and told me that I know somebody at the university, I make sure that you will not get accepted. I have to say I was not joyful about that. But in retrospect, when I was these shyest things I thought over, if he would say to me, I know somebody, you are in, I make sure, isn't that I would spend the whole summer in the pool and not studying? So that's why if I get an award, usually I thank all of those people who try to make my life difficult, because they made me work harder, more resilient. And that's what I try to tell the young one. Luckily, the book, when it was published in last year, I get letters. Somebody said that give up to PhD, get for Christmas my book, read, and went back, and he said I didn't try hard enough. And so maybe there is a positive effect. But it is difficult, of course. But, you have some cheerleaders, maybe your colleague or your husband, your children, that and help you the first shock you get when you get terminated in your position.
I read articles in the newspapers that world-famous scientists are warning before future pandemics, which will be by far more worse than COVID we had. And they say that our antibiotic culture makes the viruses more resistant against the antibiotica. And also the global warming grows up the mosquitoes and ticks in the rainforests and the permafrost, and also the diversity of animal, which will be less and less, is also a situation that the virus can grow up between the animals. And are you confident that in the future the scientists will be capable to find a vaccine, or do you have also fear of our future?
I mean, if you could read a little bit more, you would find they describe also that vaccine actually causes cancer and many other things you can read, very scary things. And considering the viruses, that the corona was easy because we already 40 years were analyzing this kind of virus, because we have coronaviruses and it is infected us, not that one, but the prior one. And we knew that the spike protein which is outside, and that's what we have to make the vaccine. And then all of the bats had many different coronaviruses which can, jump over human. So right now, for example, the trend is to make a coronaviral vaccine, meaning that aligning hundreds of different coronaviral sequences and seeing which is the conserved sequence and make a vaccine for that. This is the same for influenza. I didn't mention the advantage of these RNA vaccines is that you can have a multivalent, meaning that you can mix 100 different protein coding RNA. And right now, Moderna has RSV, the respiratory syncytial virus, the influenza, and the coronavirus vaccine with many different RNA coding, many different proteins in one shot. So that you can have, very quickly, you can develop and you can have for protecting against many different pathogens. I have to say, not in human trial, for example, the tick which spread the Borrelia, which causes Lyme disease, and also encephalitis. what the scientists did, instead of identifying what is the pathogen, they did identify the protein in the saliva of the tick, and which helps to spread those bacteria in our system. And they made mRNA coding for 19 different tick saliva specific protein, and then that's what vaccinated the mice. And immediately when the mice was bite by the tick, they immediately reacted and couldn't be infected. So might be other approaches, when even we don't know what could be the new pathogen, if it's spreading by some kind of insect, let's say. But I am very optimistic. The scientists here, a young one, and who are coming, that they will figure out.
Hope over fear.
Oh,
if
expired
it comes to
again.
now we're expired together, the both of us. But I saw that the microphone was handed, so I'm having one. Okay. Oh, a young scientist here. We have one question, and then Okay, you go first.
I would have a small question. Did your parents support you with your life choices?
Say that again.
Did your parents support your life choices? Köszönöm.
Yeah, so, I mean, I spent, when I was young, I spent time in, in a shop, in the butcher shop. So I learned that, not just making sausage, but other things. But, my parents supported my sister also, that what we wanted to study. My sister got a PhD in economy, and so she was doing well also, so that they did not wanted us to, so it could be a pressure if your father is a neurosurgeon. I talked to one, that they want their children to be, but I didn't have a pressure to be. My father was, saying that what is this researchers are doing, because he thought that, I understand he's a teacher, but what is research? And I have to say this word is an interesting word, because we are not searcher, meaning that we repeat the experiment many times. We are researcher, researcher.
In other words, you fail forward. So we have the last question.
Thank you very much for this. Is here.
Okay.
Thank you very much for this inspiring talk. I think that you started even with an approach that was not to make vaccines, but to make really other type of therapeutics. And one of the things that we learned from the technology that you really developed during the COVID pandemic is that these were products that they were very good activating the T cells, so to fight the pathogens. We have many situations, autoimmune diseases, that what we would like is to generate T cells that they do not fight, that they really are nice, as you have been telling to all of us. What do we need to change? Do you think that is the lipids, is that you need to make these bases even less immunogenic? I know that BioNTech had a paper a while ago, but I don't know if you could tell us what is the future, if you see that we could get vaccines for type 1 for autoimmune diseases in general.
Yes, so those who are not in this immunology field yet, so our body, how it recognizing something foreign is not, it is, our immune system has no eyes, no way of knowing, so that our immune system recognizing foreign things by that it is something hit them, So that in the vaccine, for example, is not enough that mRNA codes for a foreign protein. In the lipid nanoparticle, there is a molecule which hits head on the immune system and say, watch out for that and do something. And then when you want tolerization, you want somebody has autoimmune disease and you want your body to tolerate, then you need an RNA which would not activate, and you have to have some wrapping up things which will never hit head on that immune system, so that could hide. So that's what we did experiments, for example, for experimental encephalitis. In that experiment, you know, we knew, because we generated the animal, we knew what was we have to tolerization against. But many cases, the problem is that in human immune system, you don't know that what caused some autoimmunity. What was that? And many things, you know, with the immune system, I might tell you, is that there are thousands of, so how our immune system recognizes something, that we have a molecule which holds up a piece of, let's say, the virus. And thousands, maybe everybody in this room has a different holder, something in their surface. And then what they discovered is those people who ended up in the hospital because after the infection, because their holding molecule was such that from the coronavirus could pick just only two different pieces to hold up. The other is not. You understand? And some other people had a different holding molecule, and they could hold up 8 or 10 or 20 different pieces of that virus. And those people were fine. So because you don't know how your this MHC molecule, which we call this holding molecule, up until you don't know that whether you will end up very sick or you are fine. So it's no clue that you could be an Olympic champion and it won't hurt you. Yeah, you can die because you know, maybe your holder cells, holder molecule is just holding up from the whole virus just 2 pieces. Can do that. So one day, we will know our all of the genomes, and we will look at there and say, well, oh, that's the virus. Oh, I can hold up all of it. Okay, I don't need a vaccine. Or you look at there, oh my God, I have to get that because my molecule on my surface of my cells is just from that kind of virus just can hold up 1 piece, and that's not enough for me to so that we have to learn, and the technology is there. I mean, listen, if this corona would happen, it was not part of the question, but I am just saying here, that it would happen 20 years ago. 20 years ago, we could not order a gene. Today, I can order a gene for something which I think is important, and next day the gene is here. I can make an RNA in 1 hour, I can put on the cell, half an hour later the protein is made. That's how it is. But 20 years ago you couldn't order, but there are companies in different parts of the world, they start to synthesize genes, and we could order genes. So the progress, and the PCR which came, you know that you are familiar with that word, but at the end of the 80s, 89 was molecule of the year, you know, that up until that, you couldn't amplify DNA. And so all of this technological progress helps us to, and sequencing, of course, now it is $1,000 or something, but you have to understand what you see there, and it is coming. And so I am very optimistic that the future is nice. I answer all of these questions.
So I don't think we could ask for a better ending than you saying you're very optimistic. That makes me happy. We also promised that the book will be out in German on the 21st of August. We will have it in the bookshop in the pavilion for sure. We will let you know. And now it's my pleasure to invite you all for the informal part of this evening. Thank you so much for being with us.
It will be also in book, you know.